All right, welcome back everyone. Where we left off, we had just gotten into the unit of study on proteins as a biochemical type of structure, and we had just talked about the fact that proteins are based on polymers of amino acids. And there's 20 different amino acids which go into the making of all the proteins that we know of. There's 20 of them, and we can see them all here. This is sort of similar to a slide that I left you with last time. We'll go into that a little bit more in a moment, but we can see that virtually all amino acids are look the same in that we can see that if you look over on the upper left-hand side, they all have this this same type of structure on top, where we have a ionized amino group, an ionized carboxylate group. So we have an amino group and a carboxylic acid, therefore we get these amino acids. Where they differ is down the bottom in red on each of these is their side chain, and each amino acid, each of the 20 amino acids, has a different side chain associated with them, so that they're all different molecules, but they bear very strong resemblance, indeed identical structure in a lot of their structure on the top of that molecule. Let's look a little closer at one representative amino acid on the next slide, and that would be the one that we see here. Now, all amino acids have a name associated with them, and that name is based upon the architecture of the side chain that we see on here. The amino acid that we looked at, or that we highlighted in the previous slide right here, is called alanine. Alanine, there's 20 different names because we have 20 different structures here for our different side chains. Now, a couple of salient features about amino acids, the way they're expressed, some of their structure. In particular, if you'll notice the way that this amino acid, and indeed all amino acids, are expressed is if you look at it from left to right, if you sort of follow that cadence right there, we always have the amino portion on the left and the carboxylate portion on the right. So amino acids, and indeed all the proteins that make up amino acids, are always written with the N terminus on the left and the C terminus on the right. Well, by terminus, I mean end. So up here on our alanine amino acid, we see over on the left-hand side that termination, if you will, over here is there's the N terminus, and over on this side is the C terminus. I'll be talking a lot about N terminus versus C terminus, especially as a direction. And indeed, even on the level of the amino acid, our directionality will be always, or almost always, N terminus to C terminus as we look at the structure from left to right. Another notable feature about amino acids is that in all cases but one, we have amino acids are chiral at their alpha carbon. So let's look at this amino acid here. This alpha carbon that I'm putting in yellow here is the carbon in the middle. And in order to have a chiral carbon, we need 4 single covalent bonds emanating from that carbon, and they all have to terminate in 4 different environments. So here we have a hydrogen, a nitrogen, a CH3 carbon, and a carboxylate carbon. Even though they're 2 of the same element, carbon, they're in different carbon environments. So we consider them all 4 are different. So thus, amino acids are chiral at their alpha carbon. There is one exception, which we'll talk about on the next couple slides, but 19 of the 20 are indeed chiral at their alpha carbon. One last thing to note is that just as all amino acids have a particular name, in this case alanine, they always also have a 3-letter abbreviation, ALA for alanine. On most of these, Most of the 20 amino acids, that three-letter abbreviation is are the first three letters of that amino acid's name, ALA, for alanine. That isn't always the case, though. I think on maybe 3 or 4 of them that differs. So our three-letter abbreviation is somewhat different, or is different from the first 3 letters of the name of the amino acid. So we are going to explore our 20 amino acids by putting them first into categories based upon the architectural nature of their side chains. A full 10 of these amino acids have side chains which are considered hydrophobic side chain. Ultimately, what that means is that the nature of the side chain, if you look at the components in red throughout all of this, are hydrocarbon in nature. As we know, hydrocarbon is very hydrophobic. It is unlike water. So when we get a completely hydrocarbon side chain on an amino acid, or a mostly hydrocarbon side chain on an amino acid, we get the amino acid itself is considered to be hydrophobic. And this takes up a full half of our 20 amino acids. 10 of them have a hydrophobic side chain, which we see here. Some important considerations with 2 of these amino acids with hydrophobic side chains. We've talked about this already. And if we look over on the right-hand side here, on the top, we know that every amino acid has an alpha carbon, and most of the time that is chiral. There is one case in which the nature of the side chain here on an amino acid makes that a non-chiral carbon, and that is in the case of glycine. When glycine, over here on the upper left-hand side, glycine has a lowly hydrogen as its side chain. So if we look over here again on the right, we have a hydrogen here and a hydrogen there emanating from that central alpha carbon. So we have identical environments. Thus, that carbon in the center is not chiral. So glycine is the only non-chiral amino acid. Additionally, if you look in the upper right-hand corner here, any amino acid always has that nitrogen associated from it, where it gets the term amino from. In 19 out of the 20 cases, that is a primary amine. And if we look at this amine right here, how do we tell to classify that? We look at the nitrogen and ask how many carbons are directly attached to it, like right next door. And in this case, it would be 1 carbon, so that would be a primary amine. 19 of the 20 amino acids have primary amines as their amino piece on its backbone, but there is one that has a secondary amine, and that is proline. Looking down on the bottom here, just above my head, there is a proline is the amino acid where we get that side chain, and it appears to be it is made up of hydrocarbon, but it covalently attaches to that nitrogen, which is really strange. It's the only amino acid that does that. But if we try to classify this amine on this amino acid, we would see that there are now 2 carbons. We have 2 carbons directly attached to that nitrogen, so it would be a secondary amine. Proline is the only amino acid with a secondary amine. All others have a primary amine. So the next 5 amino acids, we're looking at the nature of the side chain here, and in this case we would be looking at amino acids with called a neutral hydrophilic side chain. Neutral hydrophilic. And what that ultimately means is that we have attached to that side chain something that associates well with water. And look at this over on the left-hand side. OH for serine. Threonine has an OH on it. Even tyrosine over here has an OH on it. All of these are strong, these OHs are strong hydrogen bonders. So really, no matter how much extra carbon we have attached to that side chain, that OH flips the nature of that side chain from hydrophobic into hydrophilic because of that very strong hydrogen bonder on these cases, serine, threonine, and tyrosine. Additionally, we have a couple of other ones which have, in this case, asparagine and glutamine, which have NH hydrogens attached to their side chain. In this case, in an amide, we have primary amides right here. But those nitrogens connected to hydrogens are strong hydrogen bonders. Like dissolves like. Water is a strong hydrogen bonder, so that converts these two side chains from hydrophobic, which their carbons would bring across, into hydrophilic side chains. Importantly, while these all of these side chains on this grouping of amino acids are considered hydrophilic, they are considered neutral. They are neutral. Why? Because just on the side chain itself, if we look at the side chains themselves, there is no residual charge on there. Remember, we're dealing with physiological pH here. At physiological pH, we have some components in there, such as amines or carboxylic acids, which are ionized at physiological pH, but these are not the case. All of these side chains are neutral. Now, if we look closely at our remaining amino acids, there's a grouping of 2 of them which have carboxylic acids on the side chain itself. And carboxylic acids, as we know, are ionized at physiological pH to be as their carboxylates. So these 2 amino acids which have carboxylates on their side chains are, for instance, aspartate and glutamate, have a negatively charged hydrophilic side chain. Negatively charged because of the carboxylates, CO2 minus. Hydrophilic means it likes to associate with water. This is going to be important when we stitch these amino acids together into proteins, but it is hydrophilic. Anything with a charge on it naturally is going to be hydrophilic. If you think about ionic structures typically dissolve or frequently dissolve very well into water, that's because they like associating with water. Those charges on the ions really enjoy associating with water. So this is where we can say we have a negatively charged, two amino acids, aspartate and glutamate, with negatively charged hydrophilic side chains. The last 3 of them are amino acids with positively charged hydrophilic side chains. Positively charged hydrophilic. Anytime we have a charge, we're looking just at the side chain right now, we have something that is considered hydrophilic. But in this case, they are positively charged. So if we look now at these 3 amino acids, lysine, arginine, and histidine, all have amino or quasi-amino-like functionality within their side chains. When we have that, any amines we've learned that, at physiological pH are positively charged. They've picked up a hydrogen ion. And in the case of lysine, arginine, and histidine, that side chain is positively charged at physiological pH, and thus it is hydrophilic. So we've now covered all 20 amino acids, and we've been able to sort of group them, if you will, into 4 categories. Amino acids with a hydrophobic side chain, amino acids with a hydrophilic side chain, and then the charged ones, negatively charged hydrophilic and positively charged hydrophilic. So an important consideration here, we found that a couple of slides ago that all amino acids are chiral except for glycine. If we look again now at our alanine, we have alanine right there that we talked about. Alanine has a methyl group as its side chain. We found that alanine was indeed chiral because from that central carbon right there, we have 4 single bonds that lead to that lead to 4 different environments. Now, notice in this depiction of the amino acid that we see most often going from left to right, we go N-terminus to C-terminus. And we said virtually all of the time that is how we're going to be depicting amino acids, and as well as depicting the proteins that those amino acids make up. Now, we will depart from this just for a moment in order to look at this concept of chirality and its consequence. If you remember, when we were looking at carbohydrates, in particular this very specific one. Carbohydrates we always showed in a Fisher projection. And when we put a carbohydrate in a Fisher projection, if you look up here, meant we had to put all the carbons in that zipper down the middle right there. And wherever the chiral carbon pointed out, or the chiral carbon had a hydroxyl, which pointed out, in this case, it points towards the left. That is an L carbohydrate. It's an enantiomer. If it points to the right, it is a D monosaccharide. So in these cases, we can classify these or categorize them as L or D. And we found that in nature, all carbohydrates, or rather all monosaccharides, were of the D variety. The D variety, where that hydroxyl on the chiral carbon points to the right. So the same can be said for amino acids. Now we're going to draw an amino acid or express an amino acid in a way that now departs from that almost universal N to C terminus depiction. And if we look at that, if we make we exhibit amino acids in this Fisher projection. Now this is something we haven't seen before, but for alanine, what we looked at before, all of our carbons now are in this vertical zipper right now. We see in both pairs, we have this mirror plane, we have this vertical zipper. Normally we don't express them like that, but when we do that, see here we have the carboxylate carbon, the alpha carbon, and the first carbon of that side chain. In this case, what happens is we force the amino group now either to the left or to the right. And very similar to monosaccharides, when we have the Fisher projection of an amino acid, as we're shown here, we can categorize them or classify them based upon which way that amino group points now. If the amino group points to the left on this Fisher projection of this amino acid, it is the L amino acid. In this case, it would be L-alanine. If the amino group points to the right, in this case, on this enantiomer, it is the D amino acid. Now, interestingly, nature, as we found, was very fickle. It in monosaccharides, at least, all natural monosaccharides were of the D designation. Interestingly, it does a flip-flop here, and in nature, all naturally occurring amino acids that are basically co-opted into all of our proteins are of the L variety. The D amino acids do not exist in nature, just like L monosaccharides do not exist in nature. I mean, there is some exception, however, but the vast majority of these things, there is only one particular enantiomer which is expressed in nature. Another salient feature of our 20 amino acids, the acids that go into a protein building, is they can be categorized into what are considered essential amino acids and non-essential amino acids. When we were talking about lipids, there were some essential fatty acids. That meant we had to have a dietary intake of these because animal systems, our bodies, cannot produce them. We had to have a dietary intake of them. Now, this is also the case with amino acids. Of our 20 amino acids, we have, if we can look at this, over on the right-hand side are non-essential amino acids, and we have a listing of them right here. That is about 12 of the 20, and the reason they're non-essential is that these amino acids can be converted to, we can convert to any of these 12 amino acids from certain other amino acids within our bodies. We have the biological processes which we can say convert alanine from arginine or something like that. So if we have one a grouping of one amino acid, we can convert that into the other. So these would be considered non-essential amino acids. This is in contrast to what are considered our essential amino acids, these 8 amino acids right here, where we must have a dietary intake of these because the body cannot synthesize them for themselves. So these 8 amino acids. Now, if we look at types of foods and our protein sources, the only standard protein source that has all 20 amino acids, including all 8 essential amino acids, is red meat. That would be the only one that can that has all of our essential amino acids. Now, obviously, that doesn't force us to eat red meat all the time in order to gain all of our amino acids, because we need stores of all of them to build up all of our own proteins properly. However, outside of red meat, we have to be able to conglomerate certain foods that have different protein profiles, some of which are rich in some amino acids, some of which are rich in other amino acids. So together they'll give you what is called a whole protein. A whole protein means an intake of proteins which give you reasonable amounts of all amino acids. Examples of whole proteins, if you're not a red meat fan, would be, for instance, rice and beans. Rice and beans are a sort of a vegetable combination where rice has certain high quantities of certain amino acids, and beans have certain quantities of other amino acids, and together they make up what is considered a whole protein. Couple other examples of whole proteins would be a peanut butter sandwich. There are proteins within wheat which is milled into bread, and other amino acids which are inherent in peanut butter, but put together, those two proteins can break down into amino acids where you'll have a whole protein, all 20 amino acids, or at least all the essential amino acids together. Also, an example would be hummus and pita. Both of these have stores of certain of the essential amino acids, but put together we get a grouping of all 8 amino acids. So as long as you're careful, it's quite easy to have a dietary intake of all 20 amino acids, in particular all 8 essential amino acids. Now that we've studied the nature of the amino acid, the basic structure of amino acid, the fact that we have 20 amino acids that go into making all of proteins, we find that now we get a recurring theme in nature, and it's a recurring theme that we've looked at in the biochemistry portion of this course before, that of the concept of the polymer. If you remember back in our carbohydrate chapter, we spent a significant amount of time looking at the nature of the monosaccharide, and then revealed that this monosaccharide is in fact is a monomer that can be put into polymeric form, such as dietary starches like amylose, or other polymers of monosaccharides, or cellulose and glycogen and that sort of thing. We're going to be doing the same sort of thing with our proteins. We find that the amino acid is simply the monomer, and we're going to be covalently linking these monomers together into polymers known as proteins. So really, proteins are essentially just linear polymers of amino acids. And if we look carefully down on the bottom here, we find we have two representative amino acids here. They happen to be glycine and alanine, and as you can see, they're written in their standard N terminus to C terminus form. As we look right to left, we have our amino group first, and then the last thing we have is our carboxylate group. So we put those end to end in this case, and we find that when we put these end to end, that we end up having the carboxylate component on one amino acid right up in the grill of the amino group on the other component or the other amino acid. So we have this ability now to imagine or envision a connection. So if we look, here is our reactants side of the equation, and here is the products side of the equation. Notice this reaction spits out water as a byproduct. So water, HOH, must be eliminated from the condensation of these two amino acids. So let's see if we can identify water here. We need two hydrogens. There's two of them, right? We'll take from there, and an oxygen. So if we take two hydrogens of those three and oxygen and link the rest together, we certainly over here on the right-hand side now spit out water. But let's look at the linkage that we've made here between these two amino acids. It is known specifically as a peptide bond. That is more of the colloquial term of it, a peptide bond. But in reality, it's actually an amide functional group. An amide. So let's take a look at this. If we were able to see, we discern this carbonyl group right here, which is adjacent to an NH. In this case, that was diagnostic for an amide structure. Specifically, we call that a peptide bond, though. The definition of a peptide is really a very small protein. Proteins are long polymers of amino acids, maybe 200, 300, 400 amino acids long, all linked, they're individual amino acids by these peptide bonds. A peptide, a peptide is essentially a small protein. It would be considered a protein of 10 carbons, or I'm sorry, 10 amino acids or less. So what we see right here, this is a 2 amino acid protein. It would be considered a peptide. You could still call it a protein, but a peptide is essentially a protein that is 10 or less amino acids long. So we have these linkages, and we get a dehydration where we spit out water, and this amide bond, which is considered a peptide bond, results. Now we have condensed, if you will, these two amino acids together into one covalent structure. But still, if you look over at one of the amino acids on the left-hand side, it goes N terminus to C terminus. In that direction, left to right, we encounter nitrogen first, then the carboxylate carbon second. The same goes for any protein or peptide made from these amino acids. In fact, we get, if you look over on the right-hand side, it's still expressed N terminus to C terminus. The peptide, in this case it's called a dipeptide because there's two amino acids that go into it, is still N terminus to C terminus. Whether it's one amino acid, whether it is a 2 amino acid peptide, or if it's a 400 amino acid protein that is being expressed, we always have a directionality considered in there, and that is always N terminus to C terminus, always. So in the last slide, we looked at how we would envision 2 amino acids coming together into a dipeptide, if you will, a very small protein, this dipeptide. And we could indeed extend this to 3 amino acids, 4 amino acids, 10 amino acids, 100 amino acids long. We would be able to draw this together. In this component right here, we're going to expand this number of amino acids in a peptide to 3. It doesn't have to be 3, but we're going to do that just to show that we can do this with any amount of amino acids at all. But importantly, we're going to show you there's a 4-step process for drawing these proteins or these peptides, depending on however, it doesn't matter really how many amino acids long they are, but there's a 4-step process to it. So if we were charged with drawing a 3 amino acid protein or 3 amino acid peptide, what would that look like? How would we go about doing it? Well, here's the 4-step process. And the first thing to do is to draw out 3, because we're doing a tripeptide, 3 series of NCC. If you look at up on the top here, just draw the letters NCC 3 times. And we've indeed done that right here. NCC, 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 and we have connected them all with single covalent bonds. Fair enough. So that's the basis of our tripeptide. In the next step, what we're going to take in order to show that, yes, indeed, we're going from N-terminus to C-terminus, we draw way on the left-hand side an ionized N-terminus, because amines are always ionized, NH3+ for a primary amine, at physiological pH. And at physiological pH over on the right-hand side, we're drawing the C-terminus as a carboxylate, not COOH, COO minus, because remember, both amines and carboxylic acids are 2 of our functional groups which are ionized at physiological pH. So we draw these 2 termini in there, leaving the rest for now. Third step. We're going to, wherever there is considered an amide bond, we have to exemplify that. So we're going to draw the amide carbonyls right here and the NHs on all of our, in this case there's going to be 2 of them, amide bonds. The amide bonds between the individual amino acids. There's 2 amide bonds or 2 peptide bonds here because we have 3. We're doing a 4-step, we're drawing a tripeptide, 3 series. So we draw in these carbonyls, C double bond O, and our NHs. And what that leaves last are these sort of naked carbons right here. And these are our alpha carbons. And our alpha carbons we deal with last. Now, on our alpha carbons, in all cases but one, they have a hydrogen associated with them. So we're going to draw a hydrogen on there as well in the next step, but also the side chain. And as we know, there are 20 different side chains associated with that. So here now we see which was sort of just these naked carbons up here on step 3. Now what we've done is to draw a hydrogen, and I've drawn it as an R group, but it could be anything. If it was alanine, it would be a CH3. If it was glycine, it would be a hydrogen. And we could look back at any of these amino acids and look at their side chains. I've drawn them as these big blue R groups right here. But here now, on all of these cases, we have filled in a hydrogen and that R group. Now, in the R groups, at least now that the course is remote and all of our exams are going to be open book, we can just look up the structure of that R group. Normally in this course, I would have you memorize the R groups to these amino acids, so that's a plus for you right now. With respect to these hydrogens up on top, that's the case with 19 of our 20 amino acids, except on proline. Proline is going to have, remember on our NHs right here, the proline is the only one, remember, that is that connects that nitrogen with the R group. So in that case, our NH hydrogens would be lost in this case, but that would be on a case-by-case basis. This can be done with any number of amino acids, as these 4 steps. You can have 5 amino acids, 50 amino acids. I wouldn't have you do that, but you're able to use this 4-step process in order to to finish up or correctly draw these peptides and proteins. Now that we've put, or we're working on putting these free amino acids, covalently bonding them together into amino acid sequences, essentially we're making proteins, we now have to designate, when we make these proteins, there's 4 ways of viewing this protein, 4 different levels of protein structure. These 4 levels are primary, secondary, tertiary, and quaternary, 4 different levels. And we'll start by looking at our primary structure of a protein first, a protein's primary structure. If we had to put a particular definition to that, a protein's primary structure is essentially its amino acid sequence. Importantly, written N-terminus to C-terminus, in that direction. That has meaning. Now, if you look down the bottom of the slide here, we have the 3-letter abbreviations of the amino acids in this small peptide right here. This is 8 amino acids long: glycine, phenylalanine, glycine, alanine, leucine, serine, glycine, alanine. Or given by their 3-letter abbreviations. That sequence has meaning. That sequence has meaning because if you look at this second bullet point here, this particular linear sequence ultimately determines the active form and function of the protein once it's built. If we looked at it instead in the other direction, ala, gly, ser, lu, ala, gly, phi, gly, in that direction, which would be, as we know, now we're going right to left, C-terminus to N-terminus, that would be a completely different protein. It would have a completely different function, if it had any function at all, and it's very different than the left to right reading of it. That is very, very important when we're talking about proteins. Now, the information, if we look at this sequence from N-terminus to C-terminus, from left to right, if we look at that, what determines that particular sequence? It's not just a willy-nilly random assortment of these amino acids in there. It actually has meaning. So where does that, we're going to find that particular sequence, the information for that particular sequence is actually coded within our genetic material. We're going to be looking down the road in a couple of units of study at genetics and how the information contained in our DNA actually determines the primary structure of proteins. It is only the primary structure of proteins that DNA codes for. All other different levels of protein structure are not determined by the information contained within our genetic material. So here is an example, right, of this sequence written as a model, really, and you can kind of see this peptide sequence. You can follow what I consider its peptide backbone, where we get through our carbonyls and our NHs and our alpha carbons. You can follow that throughout. I'm tracing this out in red right here. So that is considered the peptide backbone. The peptide backbone of any protein or peptide. But also on there, we're able to pick up the side chains. Here's some side chains showing here. Some are bigger than others, but there's always side chains within these. So that we have 2 different arenas, if you will, within we're looking at a primary sequence of a protein. Our peptide backbone is going to be differentiated from its side chains. So let's look to that as we go into the next level of protein structure. Looking at a representative small section of the peptide backbone of a protein over here on the left, perhaps you can see that this overall, the way it's exhibited, this peptide backbone is all single bonds. It's shown as all single bonds. So thus, you might consider that this peptide backbone would be very floppy because we as we know, around single bonds we get free rotation. Well, it turns out that the peptide backbone of a protein is not as floppy as you might think. There's a certain degree of rigidity baked into that, and that rigidity comes from the fact that it is not actually all single bonded structure, as we see over here on the left. We have ultimately what is called electron resonance hybridization of part of this peptide backbone. We saw this back in organic chemistry when we were looking at the structure of amides, in particular why amides were not as acidic like amines, and that was because in these amides we had this resonance delocalization where we get that lone electron pair keeping busy. That lone electron pair is keeping busy by donating into the carbonyl and back and forth, so that we had this back and forth piece right here. When we looked at it in the amides unit of study, we saw that, well, that was the reason for the attenuation of activity of this lone electron pair, why it doesn't just reach out and are able to reach out and grab a hydrogen ion and be the definition of acidity. That's why amides are not acidic like amines. However, this resonance hybridization, which is always happening in an amide, has a different meaning when we're talking about the rigidity of the peptide backbone within a protein. And so what we have in this case is this double-headed arrow. This double-headed arrow showed that the structure of the peptide backbone didn't look like this, and it didn't look like this. It was somewhere in the middle. And let's look at the structure now over on the right-hand side. This is one of its resonance contributors, if you will, as it goes back and forth. This is no longer a floppy, single-bonded structure anymore. There is some double bond character to it. This double bond character rigidifies it. We get a not complete, but somewhat rigidification of the peptide backbone. And it is only somewhat, because if we look at this peptide backbone, at least on the structure over on the right-hand side, we have a bona fide carbon-carbon double bond. That does not allow for free rotation, but that goes back and forth here between a carbon-carbon single bond or a carbon-nitrogen single bond, which does allow for free rotation. So what I say is since we're a hybrid in the middle here, what we have is a partial rigidity, partial double bond character, and what that does is it rigidifies the peptide backbone of any protein to a small extent, enough such that certain other types of repeating structure are able to be formed, which lead us to our next level of protein structure. Now, we had discussed our primary structure in proteins, which was essentially its amino acid sequence, N terminus to C terminus. Now, because we found that amide backbone or peptide backbone within the protein has a certain amount of rigidity to it, that allows for sometimes what we term secondary structure in proteins. Secondary structure. So now, secondary structure in proteins, if we look, what we have here down this figure down on the bottom is we have a representative protein, and these beads on the chain here are considered individual amino acids. So here we're again going to the level of the cartoon, but we have individual amino acids written here in a cartoon sense that are connected together by these peptide bonds. What ultimately happens, and notice we're going we have an N terminus over on the left to C terminus over on the right, which is the way they're always expressed because directionality has importance in protein primary structure. Because of this quasi-rigidity of our amide backbone, our peptide backbone, what ultimately happens is certain areas within a protein's primary structure start to adopt predictable and ordered patterns. And what does that ultimately mean? If we look down on the structure on the bottom, we have this meandering of our primary sequence, N terminus to C terminus. Frequently, and this is all very very representative. I hope you recognize. As we're following it along, we have areas of just random structure, but occasionally we have areas, and given by a different color here, of what is considered ordered structure. We're going to figure out what that ordered structure is in just a moment. And then it goes into an area, as we go N to C throughout this process, an area of random structure again, then perhaps going into an area of ordered structure again. That has heightened importance. And then perhaps into random structure again, until we get to the C terminus of that amino acid sequence. It is this area of ordered structure that is what is considered secondary structure in proteins. So if there are two types of protein secondary structure, this area of ordered structure, and we're going to look at them separately. The first one is known as an alpha helix, and the second one is known as a beta sheet. So let's look at those one at a time. Both the alpha helix and the beta sheet secondary structure are very stabilized structures. That's the reason they form within areas of the protein's primary structure, is because they're so stable. And the reason that they're so stable is because of hydrogen bonding between components of the peptide backbone. What we have in this drawing here is a very representative view of a protein chain. We're just looking at the peptide chain swinging around here from N terminus to C terminus, if you can imagine that. And within this structure here, this overall primary structure, we have areas in which we have these hydrogen bonding, and they're always between components of the peptide backbone. You know, the amide backbone has NHs, and it also has carbonyl groups, and we get a hydrogen bond, if you will, between those between the oxygen on a distant carbonyl and an NH hydrogen on a distant amide nitrogen, if you will. So, but due to the meanderings of the protein, or the primary structure of the protein, it puts them right on top of each other. Now, I realize this structure is very representative. But it's to show that this hydrogen bonding that we see between distant amide carbonyls and NHs is the sort of thing that happens throughout the structure. I'm showing one representative one here. However, the additive structure of this happening throughout an area of secondary structure, we get many of these stabilizing hydrogen bonds, which really act as little staples to hold these things together, highly stabilizes these secondary structures. So we'll look at this, the nature of these hydrogen bonds as we look separately at the alpha helix and the beta sheet. The alpha helix in secondary structure is the most common secondary structure type. We see it more often in proteins than we do the beta sheet. As we were saying before, the reason that alpha helices tend to form are because of this very highly stabilized structure, which we'll talk about momentarily. Now, frequently when we're showing or depicting secondary structure in proteins, there is a ribbon frequently that traces the secondary structure, in this case an alpha helix, to show within the protein to make it more visible. That obviously isn't really there in these cases, but we do show that. We'll see that going from ribbon to line to ribbon to line, and we only see ribbon in secondary structure, so it just helps us to see that. Now, in this secondary structure we have right here, given by this ribbon, we see the amide backbone. We see the amide backbone, and purposely the side chains of the amino acids have been removed for clarity. And I want to focus in on this amide backbone, and what we ultimately will see is that we're going to have areas where we have carbonyl groups pointing in one direction and NH hydrogens pointing in the other direction. Throughout this structure, we have what we saw on the previous slide of those carbonyls pointing straight at our NH hydrogens throughout this structure. And as we know, what ultimately happens in this case is we form hydrogen bonds between them. Notice, and if we look at the structure over on the far right here, we see in these dotted lines, these are the hydrogen bonds which form in an alpha helix, all up and down the alpha helix, and they're all in a longitudinal sense along the axis of the alpha helix. We see these hydrogen bonds, and each hydrogen bond in the alpha helix makes the helix itself more and more stable. They're like little staples holding together these structures, such that the more hydrogen bonds we have on there, the more stable the structure. And again, in both of these drawings right here, the side chains of the amino acids have been removed for clarity in order to show just the peptide backbone given by this ribbon structure right here. If we were to take a look down the bore, right down the bore of an alpha helix, as we can see right here by these two arrows, what we would end up seeing or depicting is the structure that I have written here over on the left, where right in here now is that bore, that central bore in the alpha helix. And the reason I show it this way is since we're looking down through the helix, in this case now a rendition of the side chains has been shown as well. We're showing these side chains sort of kicking outward from the alpha helix, sort of like spokes on the wheel of a bicycle. And that is another reason, in addition to these hydrogen bonds which stabilize the structure, it's another reason why we have this stabilized form in the alpha helix, because these big bulky side chains point outward from the helix, putting them in an area as they emit out is a more empty area, so they don't crowd together. If they crowded together, it would tend to destabilize the helix. But in this case, in all these cases, these R groups, which are the side chains, are pointing outward from the alpha helix in order to minimize sort of steric interaction between them. Now, the beta sheet is our second example of a secondary structure. Again, the beta sheet is very similar to the alpha helix, is stabilized by hydrogen bonds only between components of the peptide backbone. So here we have two separate, in this view here, two separate renditions or areas of a peptide backbone, and they could perhaps be connected from some faraway point like this. But what ultimately happens is we get these, again, these very stabilizing hydrogen bonds between the carbonyl groups on one amide with the NH hydrogens on the other. And throughout this structure, we put, here's two components of a sheet, but ultimately what happens is these things tend to zigzag on one another, and we get these continuing sheet-like structures forming. We'll show you a picture in a moment of how that ultimately works. So the hydrogen bonds are between, again, the backbone components, but they're totally distant from one another when thought of in terms of the primary structure, right on top of each other when in terms of how that geometry of the way this peptide chain or protein chain is coming together actually exists. Now, importantly, we can have two types of beta sheet, and it's important to be able to sort of follow these things along. We would have, so for perhaps an N terminus here, and as we follow this along, notice in here as we follow this along, we go to this flat ribbon structure in areas of secondary structure, and revert back to this sort of lines when we're in areas of random loops back into our our sheet and back into random, back into our sheet. So directionality is ultimately important in here. And if we look at the directionality on some of these, it depends on, we always have to know where our N terminus is and our C terminus. Notice on this top structure, the interactions here between these strands to form the beta sheet are going in opposite directions with respect to their N to C terminology. So if they're going in opposite directions but still have that very important hydrogen bonding between them, it is considered an anti-parallel beta sheet. Anti-parallel. But there's a lot of freedom in the peptide backbone, so we could imagine a case down here on the bottom where we have this sort of loopy structure going on where only the interactions are on the beta sheet. So notice these arrows now are all going the same way, and that those arrows are the direction N to C. Always we have to discern that N to C direction within a protein's primary structure, but those ribbons right now all happen to be going in the same direction. So in this case, we would have a parallel beta sheet. Both of them, in fact, do allow for what we see here on the upper structure. We got these very nice hydrogen bonds happening, but just does it happen in opposite directions, or does it happen in the same direction where these happen? The difference between parallel and anti-parallel beta sheets. Lastly, what we'll look at here is a representation of an actual protein. We can actually get sort of the meanderings of that primary structure throughout a protein, find that through certain techniques, and sort of spit this out into a computer image. And this is the actual rendition of a particular protein, but notice within it, wherever we see these ribbons within here, that's where we're depicting secondary structure. And indeed, areas within here, we see for secondary structure, we see areas of alpha helices. There can be long ones, there can be very small ones, but they would be examples of an alpha helix or alpha helices. Also in ribbon structure, what we see here now are areas of beta sheet. In this case, we have going in opposite directions here. This would be an anti-parallel beta sheet. Here we have an example of these the beta sheets going in the same direction. That would be an example of a parallel beta sheet. Throughout this all, we see examples of just random loops, and they're given not by ribbons, but just by sort of a cord-like structure throughout. So that would be indicative of areas that don't have secondary structure. Secondary structure in general it depends on the protein. Some proteins have no secondary structure whatsoever. Some proteins are almost completely comprised of secondary structure between either alpha helices, beta sheets, or combinations of both. Most proteins have at least some secondary structure within there, and that's what we see in this drawing right here, wherein we have areas of random sort of looping, but areas of this repeating sort of pattern in there, whether it's alpha helix, whether it's beta sheet in nature. That would comprise the majority of globular proteins out there would have at least some secondary structure within them.